Hey everyone, welcome. We're just waiting for things to get rolling and Dr. Offit to join us. So come on down, make yourselves at home. If there are any questions that you'd like to ask initially, then ooh, uh, if there are any questions that you'd like to ask initially, then just put them in the comments and into the live chat, and I'll try and get to those and ask him as well. So you last promotional messages. Hey. Oh, there we go. Hello.
Hey,
Hey, how are you? It's been a while, has it not?
I'm sorry, say it again.
It's been a while, hasn't it?
It
Few
has been a
years.
while, long time.
Yeah. Well, I'm really glad to have you on. We're just about to get things rolling. Thank you so much for joining me. We've got a whole bunch of questions actually that people have been sending in.
Okay. And how long does this last,
So
roughly? How long do you think will take, roughly?
I'm hoping for an hour. If you think that you need to head off before that, just let me know and we'll just cut things short and maybe we can find another convenient time later on when we can
No, hopefully
do
we
a
can
few
do
more
this.
questions.
Yeah, if we can do it in an hour, a little less than an hour, that'd be great.
Yep, that's the plan. All right, just seeing if anybody else is joining in. Of course, we are 1 minute early. So, good morning. It's about 8:30 p.m. for me here, so it's this whole time difference is really weird. Like,
This is
I had
not
this
early
chat
for me.
with
I mean,
Susan Gerbic a
I
while
was
back.
up at 5 o'clock this morning, so it doesn't matter.
Wow. I That only happens when actually that never happens. um, all right, let's get things rolling, and I'm sure people will start coming in.
Okay.
Um, Hello and welcome to the Rationable YouTube channel and the Rationable podcast. Today is going to be very special because we have a very special guest who is probably one of the most prominent voices in vaccine advocacy. He does a lot of myth busting about vaccines and how good they are for you. And he even worked on the rotavirus vaccine, which lasted about that vaccine got took about a decade to release from start to finish. So if there's anyone who knows about vaccines, it's Paul Offit, Dr. Paul Offit, who is joining us this evening to address a lot of the questions that we might have about vaccines, and especially the COVID vaccine. So, Paul, welcome. It's really good to
Thank
have
you.
you here.
My pleasure.
Right. So I want to get right down into this. I'm sure I'm sure you've been extremely busy with the vaccines being released so recently. I mean, it's in the US and in India, they've just come out as well. And things must be going kind of nuts. How are you doing?
I'm doing fine. I've had 2 doses of vaccine, so I'm feeling a lot better now when I walk
Oh,
outside that I'm not going to die. So
oh, thank
things are good.
goodness. No, it takes a little while for these things to kick in, doesn't it? Or is it just an immediately immune, superhuman kind of level of immunity?
I don't think that's true of anything, but no, the first dose offers you some short-lived protection, but not a lot. The second dose, I'd say 7 to 10 days after your second dose of these mRNA vaccines, in theory, at least according to the the pre-approval studies, you have a 95% chance of being protected against any manner of COVID disease. So the data look great.
Wow, fantastic. I honestly I can't wait till I get my shot, but there's a lot of controversy happening in India for regarding these vaccines because one of them is still in phase 3 trials but has been authorized for authorized for emergency use. But we won't get into that because I'm sure, I mean, your familiarity with that might be relatively limited, but we'll get to that a little bit later, whatever you do know about it. But first and foremost, I wanted to ask, what there are several different types of COVID vaccines, right? So which ones out are there? There is the mRNA, of course, that we've all heard about right now, and we have an adenovirus version as well. So could you just lay that out? What are the different types of COVID vaccines that are out there?
Okay, so well, I'll start from the beginning. The every type of vaccine that has ever been made before is any strategy that's ever been used before is also being used to try and make this vaccine. So for example, some researchers, especially in China, and also I think there's an Indian group that has take the virus, grow it up, purify it, kill it with a chemical, the so-called inactivated viral vaccine. I mean, that's the way we make the inactivated polio vaccine, the rabies vaccine, the hepatitis A vaccine. So that's one strategy. Another strategy is to take the virus and grow it up in the laboratory and weaken it, essentially introduce a series of genetic alterations in the virus so that it can't reproduce itself very well in you, but can still reproduce itself well enough to induce an immune response. That's the way we make the measles, mumps, German measles, chicken pox, and one of the rotavirus vaccines. That strategy is also being put forward by one of the companies, and also, well, at least two of the companies. The third strategy is to just take the key protein of the virus, in this case, the coronavirus spike protein, which is the protein that emanates from the surface of the virus. It evokes neutralizing antibodies because it prevents the virus from attaching to cells, and therefore it prevents the virus from infecting you. So you purify that protein, then you give the protein. So that's the way we make the hepatitis B vaccine or the human papillomavirus vaccine or one of the influenza vaccines. The approaches that are currently being used, such as the messenger RNA approach, the so-called replication defective simian adenoviruses, replication defective human adenoviruses, those are all genetic approaches. So now you're not really giving the viral protein you're interested in. You're giving the gene that codes for the viral protein. So your body makes the viral protein, then your body makes antibodies to the viral protein. So that's the mRNA approach. It's just a naked piece of messenger RNA that's encapsulated in like a lipid nanoparticle, a small fatty droplet, which is then taken up by the cells. The messenger RNA is then released from that lipid nanoparticle. It enters the so-called ribosomal system in the cytoplasm, is translated to a protein, and then is excreted from the cell. So you make an antibody response to that protein. And that's the two vaccines that are currently being used in the United States. By the Pfizer and Moderna vaccines, that's that. Then in terms of the other genetic approach, same idea, just bring the gene into the cell. In this case, instead of a lipid nanoparticle bringing the gene into the cell, it's a virus, essentially acting as a Trojan horse. And that's the AstraZeneca approach, was just a replication defective simian adenovirus. So it's replication defective, meaning the virus can't reproduce itself. So it can't cause disease. It just acts as a Trojan horse. And that's the same approach that's being used by Johnson & Johnson with their replication defective human adenovirus. Type 26. And those two are pretty much around the corner in the US. My understanding, I'm on the FDA's Vaccine Advisory Committee, is we'll probably be hearing about those vaccines in February, probably no later than March. And we'll see, because we need it. We need the more vaccines, the merrier. We have, just as India has, a major problem with this virus, and we need to get a lot of vaccine out there, especially when it's 2-dose vaccine. Now you're talking about doubling the amount of virus that you need for your population. So those are generally the strategies.
Yeah, I'll get to the dosage thing because that is also something that I find very interesting. But these, of course, the adenovirus vaccines, the virus that we're being infected with, which can't replicate, as otherwise it's harmless, right? We're not going to be getting infected by anything else, are we?
No, it's replication defective. It's disabled. It can't reproduce itself. Now, I mean, that's the good news. The good news is it cannot possibly cause disease, but you give a lot of it because it doesn't reproduce itself. It's really basically one virus particle per cell to be able to get that gene into the cell. So you give like 50 billion virus particles, and that's a fairly big viral load. So there are, it has just like the mRNA vaccines, it has a somewhat challenging side effect profile where you can get fever, headache, joint pain, chills. I had I've had now my second dose of the mRNA vaccine, and after the first dose, I had about 10 hours of fever and fatigue, and real fatigue. I get fatigued, but not that badly. And then the second dose was like 2 days of fever. It was actually worse than the first dose. But in theory, people, usually you see it more with the second dose, more in people under 55. I am well over 55, yet still had that response. So I see that as a good thing. Maybe my immune system is more vigorous than I thought.
Yeah, because after all the fever and the fatigue and the chills and the body ache these are all signs that your immune system is doing something. It's waking up, it's fighting against something, and in the process, learning how to fight the actual virus that it might encounter at some point. Which does bring me to the next question. Is first, these vaccines have come out in less than a year. Some of them less than a year now. I think some of maybe just about a year. The rotavirus vaccine that you worked on, that took about a decade. So,
took 26 years.
Sorry?
26 years.
Okay, two and a half decades. My goodness. Okay, so how does, why is there that, I mean, of course, we are in an emergency situation. We have to get these things out as soon as possible. But a lot of people have been asking that isn't there a danger in coming up with a vaccine that quickly? Aren't we rushing things too much? Aren't we taking too much of a risk?
Well, usually when you make a vaccine, you do preclinical trials in experimental animals, and then progress slowly through phase 1 trials in humans, which is basically dose ranging trials. You try and get the dose right, you try and get the dosing interval right to see when you can induce an immune response that you think is going to be protective against disease. And then you do phase 2 trials. So phase 1 is usually about 20 to 100 people. Then you go to phase 2 trials, which are like hundreds of people. And now you have your dose, now you have your dosing interval or doses, and you want to make sure it consistently induces the immune response you think is going to be protective and is safe. At least doesn't have a common side effect problem. Then you go to phase 3, which is tens of thousands of people prospectively either given the vaccine or not to see whether it works. Because although you think that the immune response that you've been looking at is going to be protective, you don't know that. That's why you have to do the phase 3 trials. And those usually take a few years. I mean, and involve tens of thousands of people. The difference here is that there's been sort of overlapping phases. So the government in our country have basically said, we'll take the risk out of it. We will pay for phase 3 trials. We will pay for mass producing this vaccine at risk, meaning we'll mass produce millions of doses without knowing whether the vaccine works, without knowing whether or not it's safe. We're going to assume both of those things are true. And then when we find that out, then we'll roll the vaccine out. So no company would ever do that. So that sort of truncates things. The size of the trials are the size of any pediatric vaccine trial. I mean, when Pfizer does a 44,000 person trial or Moderna does a 30,000 person trial, that's the size of any typical pediatric vaccine trial. The only difference is really length of time. So the human papillomavirus vaccine trial was a 30,000 person trial, but it was a 7-year trial. So here, the risk really isn't safety. I think if you look historically at what are the sort of bad side effects of vaccines, permanent, serious side effects of vaccines, they occur within 6 weeks of a dose. So when the FDA insisted that you have at least 2 months after dose 2, you can say with comfort that there was not going to be a serious adverse event that was relatively uncommon. Now, you don't know whether 20,000 people is not 20 million people, so you don't know whether there's a rare side effect, but you'll find that out later. In terms of efficacy, that's the difference. That's why these are approved products through emergency use authorization and not licensed. It's efficacy. You know it's effective for 3 or 4 months. But, you know, no product would ever be licensed for a vaccine that short a period of time. Well, on the other hand, 400,000 people have died in the United States this year. When you say that something's 95% effective for 3 or 4 months, and that it appears to induce memory responses, which is a good sign for longer lived protection, do you really want to do a 2 or 3 or 4 year trial before you release that vaccine, knowing 400,000 people just died? No. So that's why you move forward like this. So although I think I can understand why people worry that, you know, it's a new technology, it's been done relatively quickly, it's a sort of scary virus. You know, do I really want to get this vaccine right now? I understand the hesitancy, but the data look great. I mean, I'm on the FDA Vaccine Advisory Committee. We got a very close look at all those data, and I couldn't wait to get this vaccine.
Well, that's definitely a relief. In India, we've got the AstraZeneca virus, which is called Covishield. And that is already, and we have a huge quantity of that, which unfortunately I heard today that the company that is distributing Covishield has recently had a fire near one of their buildings. Fortunately, none of the vaccines have been damaged, and they're going to be rolling them out as usual. So nothing serious has happened. So, I mean, this is definitely, it's very good to know. But you did mention that, you know, of course, we have a dosing response. We have 2 doses that we have to take. Why do we have to take 2 doses?
Well, believe me, the companies would prefer 1 dose. It's much easier for them to do it as a single dose, and easier for the countries to do it as a single dose. When you give, so that's why you do phase 1 studies. You do dose ranging trials. You see, you know, in this case, I mean, certainly for the mRNA vaccines and also for the UK vaccines, you give 1 dose, you would get a certain level of neutralizing antibodies. And then they published this. The UK AstraZeneca group published this in the Lancet months ago. When you gave that second dose, you definitely had a higher level of neutralizing antibodies. So the thinking was that therefore that you would have a greater percent protection. That's why the second dose.
I see. But otherwise, there's no real difference between the 2 doses, right?
It's the same. I mean, well, you know, the UK story is a little how can I say this? It's just a little sloppy. I mean, when you looked at the way they did that, the AstraZeneca group did their phase 3 trial, they did it in the United Kingdom, they did it in Brazil. And they had 2 different dosing strategies. I mean, the one in the UK and Brazil, in one case, it was sort of full dose. In another case, it was half dose, full dose. In one case, it was 1 month interval. In another case, it was up to 3 months interval. In one setting, you had a meningococcal vaccine as a placebo. In another setting, you had some normal saline, salt water as a placebo. And then they sort of tried to average those 2 trials, but you can't do. You can't have 2 different dosing intervals, 2 different dosing amounts, and then try and average that out. So in one case it was 90% effective, in another case it was like 60% effective. So they said on balance it's 70% effective. We would never accept that in this country and didn't. And that's why we now have an ongoing trial in the United States with, I think it's full dose, 1 month interval, and we'll see whether or not the vaccine's effective.
Okay. And if we change that period between the vaccines, like for example, if we take 1 dose, I mean, if we change it between from a month to say 2 weeks, or from a month to say 3 months, how does that affect the efficacy of the vaccine?
That's a great question, because see, the vector is the same. The virus that's used to bring the gene into the cell is the same virus. It's this replication of vector simian adenovirus. So when you get that in dose 1, you're making a response to that simian adenovirus. You're making an immune response to that virus. So when you then get the second dose, the question is, will the immune response that you're going to make to the vector, that's the virus vector, limit its ability to enter cells and then introduce the gene that you care about? And that was the issue. And that's why they went half dose, full dose, thinking that with a half dose, they would have a lesser response to the vector. And that's why they also went sort of a longer interval, thinking, okay, maybe we'll get the immune response will settle down somewhat, and that we can get more of this vector into cells once the immune response has settled down a little bit. So that's why they did They should have all done that all in phase one rather than sort of
Mhm.
kind of fooled around with it in phase three. But we'll see what from as far as I'm concerned, on the FDA's vaccine advisory committee, we generally do studies in the United States for vaccines that are approved in the United States. So I think we want to see
Mhm.
those data in America because we want those vaccines to the trials to look like America, meaning in terms of racial
Mhm.
background, ethnic background, socioeconomic background, et cetera. So we can say to people before we give it, look, that you're represented in these trials.
Well, that is a very important thing to do. The one thing which is concerning a lot of people is that, um, especially one of the followers in Rationable, who goes by the name of Zen, he has been wondering about whether I mean, this AstraZeneca vaccine has been tested in a lot of other countries, but not in India, and yet it has been released in India, and we're wondering whether the efficacy data that it's got, the studies that it's conducted in other countries, how will that differ from the Indian thing? I mean, do we have anything to be concerned about? I mean, since we're all humans, will it probably, run the same risk and efficacy, in India as any other country? Or what do we have anything to be concerned about?
No, I think you're right. We're all humans. We're all biologically essentially the same. I mean, you can argue that where we differ is, say, in age. I mean, for people who are over 65, they have a lesser immune response in general because they're older than someone who's younger. But in terms of whether it's different racial or ethnic backgrounds or genders make a difference, they haven't in the history of vaccines. So that you don't worry about. You just want to make sure it's adequately tested. And I
Mhm.
guess in the US, we did not feel that those UK Brazilian trials were adequate enough for us to say we would license it based on those data.
Mhm. But I mean, so do you think that India should also conduct its own trials, just on the side?
You're not
Would you
going to
advise
get
the
me to
scientists to do
answer
that?
that question. I think I leave that to Indian regulatory authorities.
Yeah, because people have been wondering that. I mean, the Indian government has been very enthusiastic to get those vaccines out, and the indigenous vaccine that we have, that Bharat Biotech has released, called Covaxin, that one is still in phase 3 trials, but it's been released for use. And people don't think that's a good move. I don't think that's a good move on the whole. It's been through phases 1 and 2, which means it's safe. Am I right there?
Yeah, so that vaccine is an inactivated viral vaccine. Take the
Yeah.
virus, grow it up, purify it, inactivate it with a chemical. I know that the
Hmm.
inactivated vaccine that was being used in China was inactivated with a chemical called beta-propiolactone, which is the same chemical that's used to inactivate the rabies vaccine. I'm not
Okay.
sure what the inactivating chemical is for that Covaxin,
Mm.
but in other,
Yeah, I would have to look that up.
usually it's formaldehyde or beta-propiolactone. It's usually one of the two.
Hmm.
But the Chinese, there was a Chinese group that also developed an inactivated vaccine that claimed 79% efficacy. Then there was another trial in a South American country where they claimed that same vaccine was only 50% effective. But it really depends on how you define effective. I think the way that the Chinese were defining it was protection against moderate to severe disease, meaning the kind of
Hmm.
vaccine, the kind of disease that causes you to be in the hospital or to die. And whereas the Brazilians were also including mild disease. But frankly, from the standpoint of public health, you want a vaccine to prevent moderate to severe disease. If someone has mild disease, they're very unlikely to go to the hospital and unlikely even to seek medical attention. So you want to just keep people out of the hospital, out of the morgue. So I think that when
Hmm.
when China said 79% effective for their inactivated vaccine, that's what you care about.
Mm.
On the other
And
hand, I like to get published. I mean, you said this is like all press release, right? You really would like to see data published in a medical or scientific journal so you can really look at all that information. Because
absolutely.
we are in an age of science by press release. I mean, when the Pfizer and Moderna submitted their application under EUA, emergency use authorization, and the FDA reviewed it, and they each had their 100-page documents. We got to look at all that information. It's very comforting to look at all that information so you can see if there's any holes.
Hmm. Yeah, I'd love for more to come out, but I guess things have been so rushed. We just don't know everything just yet. Um, but speaking of which, I did want to kind of delve a little bit deeper into uh, this vaccine. It will prevent us from getting sick, but will we still get infected and be asymptomatic, or will we be completely protected from it, and therefore we won't pass it on to our loved ones if we do get infected somehow?
Yeah, so there aren't that many vaccines that provide what is called sterilizing immunity, meaning that you're not only protected against disease, you're protected against infection, meaning asymptomatic shedding of the virus.
Mhm.
Measles vaccine does that, but most vaccines don't. The pneumococcal vaccine still allows for asymptomatic infection and shedding. So does the rotavirus vaccine. So does the pertussis or whooping cough vaccine. So does the influenza vaccine. So that's the usual story. But remember, it's not binary. I mean, when you're asymptomatically infected, if you've been immunized, as compared to you haven't been immunized, you're usually shedding less virus, so you're less contagious. And the fact is, for the rotavirus vaccine was introduced in the United States in 2006. We've basically eliminated rotavirus from this country, even though it really doesn't prevent asymptomatic shedding. But I think it lessens the amount of shedding. So we were able to get on top of that virus. And I think that's what would happen here too. We make a lot about this issue of asymptomatic shedding. And I think, we ask people to wear a mask and still physically distance, even if they've been vaccinated. But that's probably not going to have a big public health impact. I'm now vaccinated. I think even if I were asymptomatically infected, I'd shed much less virus. So I'll wear the mask. I'll still physically distance because, first of all, the vaccine is not 100% effective. No vaccine is 100% effective. I still have a 1 in 20 chance of getting disease if I'm confronted with this virus. So I would wear the mask for that reason. And I think I'll feel better about not wearing a mask and physically distancing when we've really gotten on top of this virus.
Yeah, and everybody seems to have had the vaccine. But do you think that this is? I mean, I don't know if there's any data out about this yet. The last time I checked, this was kind of inconclusive. That whether this is going to be a long-term vaccine that it will keep the virus at bay for a very long period of time, or will this be more like a flu vaccine, which we'll have to get every year to ensure our immunity? Which way do you think the COVID disease is, or rather the SARS-CoV-2 virus is mutating? Which way will it be?
Right. So the SARS-CoV-2 is a single stranded RNA virus. Like all RNA viruses, it mutates and creates variants. That's true of all RNA viruses. And so measles is an RNA virus. So is flu. Flu is an RNA virus that mutates so much from not only from 1 year to 1 year, but from over minutes. I mean, this virus is constantly moving. And as a consequence, natural infection or immunization the previous year doesn't protect you. So you need a yearly vaccination. We've had a measles vaccine since 1963, and that virus has never mutated away from the vaccine. It's been 60 years, more than 60 years. So I don't. The question is, which of these two viruses is this going to be like? Is it going to be like flu, or is it going to be like measles? I really don't think it's going to be like flu. I think it's going to be more like measles. And as a general, coronaviruses are slow mutators, unlike flu, which is a fast mutator. We just need to keep looking and see whether or not the virus starts to drift away from the immune responses induced by the vaccine. And that's what's most important for me right now, just in terms of talking about these variants. If someone in our country has been fully vaccinated and then is admitted with severe COVID-19, severe infection, we need to isolate that virus and see whether or not that virus has mutated away from the vaccine. Now, I mean, again, the vaccine's not 100% effective, so it may be that it's the same non-variant virus that's just causing disease in somebody who just isn't protected, or it may be that person's not very good at making an immune response, whatever the answer is. We just need to make sure that we are not seeing the emergence of a variant virus which resists immunity from vaccination. And that's number one. And then if that's happened, then we need to do everything we can to isolate that variant. And assuming it's hard to isolate these viruses, we're going to need to also include that in a vaccine strategy. I hope that doesn't happen. It's going to be then essentially a multivalent vaccine. So it's not just, for example, the messenger RNA that's just from the non-variant strain. It'll also be the messenger RNA from the variant strain. So it'll be more than one messenger RNA, or more than one, replication defective adenovirus strain.
And so right now, it seems well, we do have a couple of different strains which have come out. So there's the UK strain, there is a South African strain. Are both of these, have they mutated enough to make the vaccine useless?
Well, so supposedly for the UK strain, the B117 strain, that according to the people at Pfizer, when they take the sera that were generated following natural immunization, those sera neutralize the UK virus. So it is their point that that virus has not mutated away from the vaccine. With regard to the South African strain, it looks more worrisome. The study that, and these are all sort of preprints, or you're trying to find data on Twitter. It's really not easy to get data, but if you look at the preprints that I've seen, about 3 that have come out over the last few days, it looks like there is, when you look at convalescent sera, people who have gotten the infection and have survived, that only those sera which have very high titers, high levels of neutralizing antibodies, still neutralize this variant virus. Now, as a general rule, the vaccine generally does better than natural infection. I mean, as compared to natural infection, after your second dose of vaccine, you're usually sort of in the top quartile, or at least top half of the sort of range of titers that are neutralizing titers that are induced by natural infection. So immunization may be better than natural infection. So the data you really want are, are these variants neutralized by sera generated from natural infection or from immunization? Are these variants neutralized by sera generated from immunization? Because people spend a lot of time working on monoclonal antibodies and trying to sort of map out the epitope that's now, gone away from the vaccine. I don't care about the monoclonal antibody studies. Just give us the practical answer, which is, I've been vaccinated. Do my polyclonal sera recognize and neutralize the virus? That's what you need. And the problem is, right now, we're depending on the companies to do that, because the companies are one that have that sera. So what I think has to happen is, people who've been immunized, get 100 people, 1,000 people who've been immunized, and get them to give your plasma, so that you can then distribute it out for a number of, say, research laboratories. And whenever these variants come up, you can look at those sera that are generated from immunization and see whether they neutralize. Those are the studies you need, and you need them immediately when these variants are detected.
Well, let's hope they come out soon. Could you just, for our viewers, explain the terms you've used: polyclonal antibodies and sera, etc.?
Yeah, sorry. So viruses have, in this case of the SARS-CoV-2 virus, the protein you're interested in is the spike protein. That's the protein that attaches the virus to cells. In that spike protein, there's one domain called the receptor binding domain. That's the business end of that particular protein. That's the part of the protein that binds to the cell. So when you make a vaccine, and you immunize somebody with a vaccine, they make antibodies to that receptor binding domain, which has a series of sort of distinct immunological components called epitopes, many epitopes. And so some of those epitopes might change so that they're no longer recognized by antibodies. But the antibodies that are directed against one epitope are generally monoclonal, I mean, just derived from one clone. Whereas if you look at all the epitopes, all the different distinct immunological regions, all those antibodies are considered polyclonal, because it's more than one clone that's generated the antibodies against all these different sites. So researchers use monoclonal antibodies. So for example, Donald Trump, when he was in the hospital, got a monoclonal antibody preparation. It was a cocktail of a few monoclonal antibodies directed against that receptor binding domain, which enabled him to survive and watch Joe Biden get inaugurated.
Congratulations about that, by the way. We are all heaving a huge sigh of relief across the world.
Thank God.
Yeah. End of an error, as is the viral term that's going out right now. I did want to talk about a little bit some of the scary news. I think it was in Norway that several people have died after getting the vaccine. Do you know what's going on over there?
So it's hard to tell. Again, it's like reading the tea leaves, because you're not getting all the details of those clinical stories. The sense that you get, it was a super elderly, meaning sort of people over 80, who, when they got this vaccine, had the side effects that are typical for the vaccine, which can be not only fever, but can be nausea, vomiting, even diarrhea. And you wonder, what their medical problem was that meant that when they then had those symptoms, it sort of put them over the edge so that they were unable to handle it. That kind of challenge to their system was more than they could handle. We haven't seen that here, and we've certainly vaccinated older people. So I'm not sure what happened in Norway. I don't know. I can tell you what scared people, though. So I got a rash of phone calls or emails from people saying, I'm 82, should I get this vaccine?
Well, I'm really glad that this might be more of an aberration than a trend. The government has issued, the Norwegian government has issued some sort of warnings, but we'll just have to see how that plays out, and hopefully everything's all right. But as of now, everybody should know that at least that the Pfizer vaccines are good. They have been working in other places, and a lot of people have benefited from them. We shouldn't be scared about that just yet.
Right.
Um,
Be
Adarsh
scared of the virus.
asks, sorry,
We should be scared of the virus.
of the virus, absolutely. Adarsh has asked a question. There's so many vaccines out now. In Dubai, one can choose any one of the two available. How can one make an informed decision about these with so much information floating around? What are the things that we should focus on?
Well, you want to make sure that the vaccine is effective enough. I mean, so for example, we have two vaccines in our country that are both essentially 95% effective, and they're highly effective in people who are older, and they're highly effective across a variety of racial and ethnic backgrounds.
Mhm.
So there's really no distinguishing between them. Let's suppose the next vaccine comes out, the Johnson and Johnson vaccine, and it's 75% effective. Let's assume that's true. Now, these vaccines, the mRNA vaccines, were 95% effective against mild to moderate severe disease. If the Johnson and Johnson vaccine is, let's say, 80% effective, the critical question I would ask is, what's the percent efficacy in people who have moderate to severe disease? Because I imagine that would be generally high. And that's good enough. And also, it's likely to be a single dose vaccine, which is an advantage. And we also have commercial experience with the Johnson & Johnson vaccine because it's the same vaccine that was used to eliminate or get on top of the Ebola outbreak in West Africa. So a lot of people have
Mhm.
already gotten that kind of vaccine. Whereas,
Okay.
mRNA, we're still, in theory, going to learn about because we haven't used that vaccine before. So there's always all these different factors. My attitude on these things is that, get the one you can get right now
Mhm.
because these vaccines are in short supply.
Absolutely. Tushar asks, can governments make vaccines compulsory, or vaccinations compulsory? And should they make it compulsory?
Yeah, I don't think we can do that. These vaccines are not licensed products. They're approved through use under emergency use authorization. Um, that's the same thing as basically giving permission to use an investigational new drug. That's really what emergency use authorization is. So I think given that, I don't think you can fairly mandate a product which is basically being permitted for use as an investigational new drug. I don't think you can do that. Now, it may be that the companies will go back, they'll submit a biologics license application, it'll be a licensed product, and we're going to have, the disease is still rampant, and then the question becomes, if you're going to walk into a classroom, should you mandate that a teacher get a vaccine? If you are going to be taking care of patients in a hospital, most of them obviously aren't going to have this virus. Should you be mandated to receive a vaccine, knowing you're also taking care or might be taking care of patients with the disease? Well, this is what we do with flu. I mean, the flu vaccine is mandated in our hospital. So then it
Mhm.
becomes a different story. But I think right now, under emergency use authorization, I don't think you can do that.
Well, fair enough, because there is a lot of vaccine hesitancy, and of course, the vaccine hesitants and the anti-vax activists are, of course, out in the hordes, uh, saying that we shouldn't get it. And I'm sure there are a lot of conspiracy theories out there about the kinds of effects that the COVID vaccines have. What are the slightly more out there or strange conspiracy theories or claims that you've heard, that you've encountered?
Right. Well, see, the anti-vaccine. I mean, first of all, I would divide this into two groups. The first group, which I think is a perfectly reasonable group, are vaccine skeptics.
Mhm.
They're worried about how quickly this has been made. They're worried about the fact that it's a new technology. They want to be reassured that this is a safe and effective product, safe more than anything else. That's okay. I think those people are generally reassurable. I just think you have to find out what they're nervous about and then answer those questions. The second group, and this is what you're alluding to, are the more what I would call the vaccine cynic. I mean, these are people who believe there's a conspiracy. You're part of it. They think that the pharmaceutical companies control everything, and that the government's lying to them, and that the medical establishment's lying to them, and the pharmaceutical companies are lying to them. So there is no convincing them.
Yeah.
I think Neil deGrasse Tyson says it best. He says, if someone reaches a conclusion without using logic or reason. Logical reason is not going to talk them out of it. So I think that's what we're here for. The crazy ones that I hear, and it's interesting, the conspiracy theorists always have the craziest notions. I mean, they're like always wrong about vaccines, these anti-vaccine activists, because they're conspiracy theorists. So the ones are, that the vaccine contains a microchip, and that, it's just a way for the government to sort of find out where you are and control you. First of all, the microchip would not fit through that needle, but in any case, that Bill Gates,
of course, Bill Gates can build it though.
Right. So Bill Gates, so he's sort of blamed for this, that he started the pandemic, that he's making money off the vaccines because I guess, $60 billion was not enough for him. So he's, trying to make money off this, which of course is complete nonsense. That the vaccines can cause infertility, which is not true, actually. I mean, there's been about 30,000 cases of women who were pregnant when they got infected with this vaccine, and there's not any evidence of an increase in incidence of miscarriage. So the natural virus doesn't seem to do that. There's no reason to believe the vaccine would do that. What else? That the virus, because it's a genetic vaccine that can alter your DNA, which is clearly not true. I mean, it's messenger RNA. It's not.
This is one of the first things I heard about.
Well, it's genetic. It's genetic. You're being inoculated with a gene, so it's not crazy for people to think that you can that it would somehow affect your genes. But it's not. In order for messenger RNA to affect your DNA, first it needs to get into the nucleus, and the nucleus has a membrane. So, and in order to get into the nucleus, you need a nuclear access signal, which this doesn't have. Even if it was in the nucleus, it's RNA. It's not DNA. It's a different language. It's like Spanish and English. It's two different languages. In order to be like DNA, it has to be converted to DNA via an enzyme called reverse transcriptase, which this vaccine doesn't have. So it can't be converted to DNA. Even if it was converted to DNA, which it can't be, it would have to be integrated into the DNA, which requires another enzyme called integrase, which it also doesn't have. So there's 3 reasons why it could never happen. I mean, the chances are not just small, the chances are 0 that it could ever happen. I mean, you have a better chance of getting this vaccine and developing X-ray vision than you have of having it alter your DNA.
That's a great analogy. Okay, we've got another question from Molesh, who says, how is herd immunity different from this vaccine for a disease like polio and for COVID, as both have different routes of transmission?
Right. So you can induce herd immunity by vaccination. I mean, that's what we do. That is the way to induce herd immunity. A natural infection is never a way to induce herd immunity. You can no virus has ever eliminated itself because it caused enough people to be naturally infected that the virus disappeared. That's never happened, and it wouldn't happen here. Only vaccination eliminates viruses, and the degree to which the percentage of the population that needs to be vaccinated in order to induce herd immunity depends on 2 factors. One, the contagiousness of the virus. The more contagious it is, the larger percentage of people that need to be vaccinated. Measles, for example, is highly contagious. Measles is 20 times more contagious than this virus. Yet we basically eliminated measles, and we did that because we had an incredibly effective vaccine. The second thing is vaccine effectiveness. The more effective it is, and frankly, the more long-lived that protection, then the fewer percentage of people that you need to immunize. So there's actually a formula for this. If you plug it into the formula, given what we know about this, the vaccines and the contagiousness, you'd probably need to vaccinate about 60 or 65% of the population to start to see a clear stopping of the spread. Now, remember, in our country, we report about 25 million people as having been infected. That's what they say. If you look at CNN, it always says like 25 million people infected, 400,000 people died. But those people that are said to have been infected have been tested and found to be infected. Many people who've been infected have never been tested. So when they do antibody surveillance studies to see who has antibodies to this virus and who doesn't, before there was a vaccine, you could see that many more people have been exposed than you would know from just those who've been tested. It probably off by a factor of 3. So it's probably more like 70 million people in the US have already been infected, which is already 20% of the population, which means 20% of the population essentially is already immune, at least to this non-variant strain. So I think we're building on that 20%. And so if we can get another 60% of people or so immunized, which will mean, I think for us, inoculating about 2.5 to 3 million people a day, we could probably get there by the summer, assuming the variant viruses don't. Change.
We've got a much longer way to go since we have over almost 1.5 billion people in this country. That's going to take a while. But I wanted to ask something. I think Zen had also sent another message, another question of whether this concerns the mutation of the COVID, of the SARS-CoV-2, which is the COVID virus. What are the chances of COVID becoming like HIV in the sense that it gains the ability to hide from the immune system?
No, that's not this virus. I mean, this virus, there's the viruses that really hide from the immune system. Well, HIVs do. HIV paralyzes the immune system. That's what it does. I
Mm,
mean, it reproduces itself in T cells, which are kind of the orchestrator of the immune system. T cells help B cells make antibodies. T cells help cytotoxic T cells kill virus infected cells. And the virus continues to mutate. I mean, you make an effective immune response to HIV when it first infects you,
mm,
but then that surface of the virus constantly changes. So whereas flu, for example, will mutate from one year to the next, human immunodeficiency virus mutates and mutates and mutates during a single infection. That's why you never get on top of it. There are viruses that hide in your body, but those are generally the herpes viruses, which is, say,
mm,
herpes simplex virus, chickenpox virus, Epstein-Barr virus, the virus that causes mono. Those are the viruses that live latently, silently in your nervous system or in your body. But that's not this virus. That's not coronavirus.
So, but what is stopping the coronavirus from becoming like those other viruses? What are the evolution processes of viruses that kind of that that make sure that it kind of follows a certain path? You would say I'm not sure if that's the right analogy, but what is it that how do viruses evolve, and what would prevent it from becoming like another virus or developing the same mechanisms as another virus?
Yeah, it just, I guess that's just the historically, it's not the way it works. I mean, um, polio viruses have the capacity to reproduce in the intestine and then travel into the bloodstream and go to the central nervous system. Measles virus can do the same thing. It can enter the bloodstream and then can travel to the nervous system. Influenza virus doesn't do that. Rotaviruses don't do that. It usually has to do with receptors, binding receptors, where the virus can
Mm,
only bind to those cells that essentially let it in. And that's probably the biggest reason. They also have different sort of strategies in terms of how they enter cells and what happens when they get into cells. They're just dramatically different viruses. Herpes viruses generally are much bigger than a virus like coronavirus. Coronavirus only has 4 structural proteins. I mean, you look at something like chickenpox has like 70 structural proteins. It's a little virus. It's like hepatitis A virus. It sort of
mhm,
just generally gets in and gets out. It doesn't do
mm,
anything. It's it, 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 we've identified coronavirus as human coronavirus since the 1960s, and they're pretty consistent. I mean, every year, probably 15 to 20% of the viruses that come into our hospital, winter respiratory viruses or coronaviruses. We see that every year with the human coronaviruses. This is a
mm.
novel coronavirus. It's not a human coronavirus. It's a bat coronavirus. So it behaves differently.
Okay, so I have an analogy for this from my understanding of it. It's like wondering how, say, um, how a giraffe can evolve into a tiger. Would that be would that be an accurate or even close to accurate analogy for this kind of mutation?
Yeah, no, they're dramatically different, probably more different than a giraffe and a tiger. I mean, at least both of which all came from sort of similar life form. These are just dramatically different viruses in how their strategies to gain access to people and survive in people. I mean, the perfect virus is a common cold virus. Doesn't kill you. It's easily spread. Um, and that's the advantage of the virus. It's never to the advantage of the virus to kill you or make you very sick. They need you. The virus needs your cells to keep itself alive, and
Yeah,
it wants
and
to
there's
be it wants to
never
be able to go from person to person easily.
yeah, and it's not like they have an intention to mutate in a particular way to avoid certain systems or defense mechanisms that the body has. I mean, they're just evolving randomly. It's just natural selection of what gets picked off and what survives. Um, but there would be a real danger. I mean, I wouldn't say this is from the COVID virus. I mean, of course, the new strains are becoming more infectious, but they're not necessarily more deadly, and they are also, uh, at least the current vaccines can deal with some of the new viruses, new strains appropriately. But I've heard this on the Skeptic's Guide several times that the COVID pandemic is only a precursor. It's a trailer to what could happen, which is where we have a virus that is maybe just as infectious, but twice as deadly, or has a fatality rate much, much higher than what COVID has. And we should feel kind of fortunate. But what is it that we should be doing as a planet and each individual nation, or even as individuals, to prepare ourselves for such an eventuality, and as we go forward?
Well, hopefully this has been a painful and dramatic lesson. I mean, when we had
This is no
the
joke.
we've had, we had SARS-1, which sort of raised its head in 2002. But, for the most part, it was gone by 2004. It really just caused moderate to severe disease. There was really very little or no asymptomatic infection and transmission. So it was easy to put a moat around that. MERS also sort of came up about 10 years later and kind of perks below the surface, but not badly. Again, same reason. It's really, isn't spread asymptomatically easily. That's not true of this virus. This virus obviously spread asymptomatically and it's devastating. When H5N1, the bird flu, raised its head, around 2005, there was put in place, at least in the US, a pandemic preparedness plan. Here's what we need to do. Here's how we're going to make face masks. Here's how we're going to make personal protective equipment. Here's how we're going to try and use the Defense Protection Act to compel companies to make vaccines. Here's how we're going to do all that. And basically it was thrown away. I think when the Trump administration came in, they just threw it away and they decided to just deny the existence of this virus. Let's just pretend it's not there. We'll close our eyes really tightly and hopefully it'll go away. That was their plan, basically.
Like magic.
Right, like magic. And they were hoping things like hydroxychloroquine or bleach or UV light or oleander leaves, pushed by the My Pillow guy, that would all change things, but it didn't really work out that way. So we did make a vaccine, for which they get a lot of credit. However, now that the Biden administration has taken over, they realized there was actually no distribution plan. There actually was no distribution plan in place. That was today's news. So in any case, I think we have now learned, as far as I'm concerned, permanently, given the level of economic devastation and joblessness and homelessness and food insecurity, how this has brought so many countries to their knees, that when a virus starts to spread in a particular part of this world, we are all at risk. I mean, I think that ophthalmologist in Wuhan, who was the whistleblower, was a hero, but we shouldn't have to have had depended on a whistleblower to tell us that there was a pneumonia that was killing people in Wuhan with the novel virus. I mean, and by then it was really too late. It was all sort of had distributed itself out. So we need great international surveillance systems for when these viruses raise their head. When they raise their head, we need to be able to put in place very quickly mass testing systems, which South Korea did, which Japan did, which we didn't do. And so we had a much bigger, I think we had more people die yesterday in our country than has died in South Korea in a year or in Japan in a year. That's how bad we were at this virus.
Yeah.
We're a bigger country, but I don't think that's the only reason. I think we could have been much better at this. We just weren't ready to do this.
Yeah, I think as a planet and as a people and even as individuals, we just need to be mentally prepared for something like this happening within our lifetimes. This is probably a once in a lifetime occurrence, but within our lifetimes, we shouldn't just be complacent about it. We should, as nations as well, make sure there are measures in place that can take care of these things. Okay, we have one more question. What is the current status? This is from Anant. What is the current status of immunogenicity? Immunogenicity. Okay,
you got it. Immunogenicity. You got it.
Okay. All right. Cool. Excellent. Okay. Immunogenicity of the vaccines being developed across the world against the new strain of COVID-19 from UK. I think we've kind of addressed this.
Yeah, so look, the UK strain doesn't look worrisome. It looks like at least the Pfizer people had said they had used their sera that were generated from people who were immunized with Pfizer's vaccine and found that it efficiently neutralized that virus. Good. That's what you want to know. But I think we need to be prepared for the fact that not all variants may be like that. And that if there's one that really is different, that we have to at least get ready to make a vaccine that contains just more than the one strain, the non-variant strain, but includes the variant strain.
Yeah. And let's hope it's not like the flu virus and have it nice long immunity to it. Oh, Zen says we need a virus defense force. It's like Space Force, but for
I like that.
viruses. We should definitely. But thank you. I think that's all we have time for at this point.
Great.
And as if more questions come in, I'll compile all of them and I'll ask you when you have time next to come and join us and help us out with that.
Okay. That sounds
But
great.
in the meantime, I would like to first say that you've already written two fantastic books. Uh, is this one signed? I've got one signed copy, I think, from you. But either way, for all the viewers, there is Bad Advice by Dr. Offit and Do You Believe in Magic, which is vitamins, supplements, and all things natural, a look behind the curtain, which is next on my reading list. And you've recently come out with another book, isn't it? Overkill?
Yeah, when modern overkill, when modern medicine goes too far. It's all those situations in modern medicine where there's abundant scientific evidence that say we shouldn't be doing something, but we do it anyway. Because I thought what
Well,
I was sort of alienate the few remaining people that actually liked me. That's what I thought I would do with this book.
Well, I read Bad Pharma recently by Ben Goldacre, which was eye-opening. Which, I mean, I've been reading a lot about alternative medicine, etc. But reading about the actual the medical infrastructure across Europe, the UK, the US was eye-opening, and there are plenty of flaws out there which need to be addressed for sure. So I'm looking forward to reading that too. How else can people get in touch with you or read your or find your books? How can people find out more about you?
There's a website which is paul-offit.com. See, I never remember this. My wife sets this up, but it's paul-offit.com. I'm pretty sure that's what it is.
Okay, I'll put it in the description.
Okay, sounds good.
And are you on social media somewhere?
I have Twitter. I do at Dr. Paul Offit. I do that.
All right, I've already tagged you, I think, in the post promoting this. But Dr. Offit, thank you so much for joining us. You've cleared up a lot of questions. I think a lot of people have been asking, and I have been asking too some of these. So it's been great to have you on. Thank you so much, and thanks for joining us. Hopefully, we'll talk again really soon.
It was my pleasure. Stay safe.
You too.
All right. Bye-bye.
Bye. All right, guys, thanks so much for tuning in. This has been a very enlightening session. I think I've been wanting to have Dr. Offit on for a very long time. I met him in SciCon in 2018. That's where I got these books actually. But I mean, he is a treasure trove of information when it comes to vaccines. And he's probably one of the most vocal promoters of vaccines around that at least that I have seen, who has been, I mean, of course, there are lots of medical influencers and social media influencers and pro science and skeptics who have been promoting the use of vaccines and getting, shooting down bad ideas when it comes to vaccines. But Dr. Offit is definitely on the top, and he's unfortunately faced, borne the brunt of the hate that one gets when one is in that position. But I hope you guys are doing okay. Thanks so much for joining this live stream. Of course, this is going to be up everywhere. I'm going to put it up on the podcast at least over the weekend because I think this information needs to go out to as many people as absolutely possible. And I just wanted to let you guys know that I'm coming up with a few new things over the next couple of weeks. So stay tuned for that. The frequency of the podcast and the articles and more interviews coming up, which I've got scheduled about the COVID vaccines. I am trying to put them together. I haven't gotten any confirmations yet, but I'm working on it. So stay tuned for that. There'll be more videos, more live streams, more interviews, so we can get to the bottom of this. In the meantime, you know where to find me. I'm on the Rationable Conversations group on Facebook. I'm on Twitter at BeRationable, Instagram at BeRationable. And you can write to me at abhijit@berationable.com or just visit berationable.com and leave a comment, read the articles. I hope you enjoy that. And I will be seeing you all very soon. Thanks again for joining me and Dr. Offit in this discussion. I will be seeing you really soon. Stay rational. Bye.